So I guess it's time to get started with our next lecture. So the next lecture is on visualization, exploration, and analysis of biomedical volume data. The lecture will have two parts. The first part will be given by Stefan Bruckner, and the second part by Michael Krone. We will have a short answer and question session in between and a longer session in the end. So I will introduce both of the speakers now already. So the first speaker is Stefan Bruckner. So he is a professor in visualization at the Department of Informatics at the University of Bergen in Norway. And so he has a broad research interest in developing methods for the analysis of complex data. The current research interest is also the development of interactive approaches for large-scale heterogeneous data spaces and data-driven science. So with his research, he aims at first to supporting scientific discovery, but also medical diagnosis and treatment, and science communication to the general public. So before moving to Bergen in 2013, he was an assistant professor at the TU Vienna in Austria, where he also received his PhD in computer science. The second speaker is Michael Krone. Maybe everybody knows him from his work in medical visualization. So he is currently a junior professor for big data, visual analytics, and life science at the Department of Computer Science at the Eberhard Karls University of Tübingen in Germany. So before moving to Tübingen, he was a postdoctoral researcher at the Visualization Research Center of the University of Stuttgart, and also a guest lecturer at the Ludwig Maximilian University in Munich. So he is also a member of the Interfaculty Institute for Biomedical Informatics. His research interests include visualization, computer graphics, GPU computing, and human-computer interactions in general, but altogether with a strong focus on molecular visualization and visual analysis of biomedical data. So with this, I would like to give the word to Stefan and invite him to start with his part of the lecture.
Yes, thank you very much for the nice introduction, Ingrid, and thanks, of course, to all the organizers for making this very nice event possible. And today, in this lecture, we have been tasked into telling you something about the fundamentals of volume visualization, and we thought we split this up between the two of us roughly into two parts. So first of all, I will really try to convey the basics of volume visualization. So even for some of the participants who have maybe not had to do much with volume data, I will try to give you sort of the foundations of this type of data and visualization approaches for this type of data, and then later, Michael will focus on more advanced application in various directions within the sphere of biomedicine. And with that, let's get started. And actually, Helvig's great talk earlier on was already a very good setup for what we will be looking at here, since, in fact, quite a few of the examples were from the realm of volume visualization or volume data in general. And one of the reasons for this is simply that when we want to investigate the world around us, and in particular the way life works, then by its nature, it's three-dimensional. And when we really want to figure out all the details, we want to look at all of these three spatial dimensions. So this is why, in particular, in a biomedical context, volume data is of interest. And in general, we can think about volume data in multiple ways. So one way to think about it is just a distribution of values over three-dimensional space. So for instance, we could look at it as some sort of point cloud where these points have hold particular quantities and they influence their surroundings in some way. And it's a perfect valid way of looking at this. Maybe in particular, again, in the biomedical context, what is somewhat more common is to think about volume data as sort of a cube, or rather as a stack of two-dimensional images. For simplicity, we can think about them as grayscale images, as you see here, and we're stacking up these images, and now we have in space these gray values, and we can already see if we now, for instance, draw a contour that connects all the pixels in those images with the same gray value, and we do this on every slice on every one of these cross-sectional images, we can see, oh, we're sort of spanning up a surface and in 3D. And obviously, if we now choose another data value, there will be another surface in 3D. So looking at this as a stack of slices is in fact quite common. However, for many purposes, it's actually much more convenient to look at this type of data as a continuous field, in the same sense that physicists talk about fields. So there we don't really say we have discrete slices, but we rather think about this as a function over the three spatial coordinates, defines a quantity in principle at every point in space. This can be established in multiple ways that I will not go into detail, but for instance, if we have such a stack of slices, we can define an appropriate interpolation function that will generate such a continuous field from our volume data, even if they are only sampled at discrete points in space. And if we do this here now, well, then we will maybe see something like this. So we will see some sort of cube, but of course, that's not really what we usually want. We usually want to actually see inside the data in some way. So we could, for instance, now associate a particular transparency with each of these data values. And then we can actually start to really look inside the data and, for instance, create this kind of semi-transparent 3D visualization from our original volume data. So that's sort of the pipeline that we will be looking at in a bit more detail here today. Let me just briefly also talk about where do our data come from. There are other lectures in this week already that will do this in more detail, but just to give you some intuition and also to briefly show that, of course, albeit a big application area, medicine and biology, there are many other application areas, also not just the four that are listed here. So in medicine, for instance, we have multiple imaging modalities that produce volumetric data: X-ray, computed tomography, magnetic resonance imaging, but also so-called functional modalities that also give us information about the physiology, such as positron emission tomography, and even live modalities such as 3D ultrasound, where you are actually creating 4D data in a sense, because we are creating 3D data at many frames per second nowadays. Those are common sources for medical volumetric data. In biology, there are also various kinds of microscopic modalities, confocal microscopy, light sheet microscopy, but also there is many different variants of optical sectioning approaches where you're actually taking a specimen and you're usually carving off, for instance, thin slices, and then you're actually imaging those slices optically with a very high resolution camera or a microscope for that matter. And very clearly in biology, this is more feasible. Usually in the medical area, this is not commonly done for obvious reasons. But of course, in engineering, we have many of the same types of imaging modalities. There are industrial computed tomography and industrial ultrasound modalities. But of course, there's this the whole area of computational fluid dynamic simulations, for instance, in the aerospace or automobile industry, very important. And where you are simulating multiple quantities over space, such as pressure, temperature, and so on. And similar approaches exist in the earth sciences, climate, weather simulations, but also, again, optical imaging. So this was just very briefly to give you a sense where do our data come from. So now let's look at how we can visualize such data. And maybe the simplest thing that we can do is to visualize our data as individual slices, which was in fact the earliest thing to do. And nowadays, this is not really any more done, but it was quite common that even though you acquired three-dimensional volumetric data, radiologists would, for instance, look at the actual radiographs on a light box. Nowadays, this is done by the means of a radiology workstation where you can scroll through the slices, and you have some means for adjusting the contrast of the slices. That is usually referred to as window level adjustment. We will actually see this a little bit later. And again, we think about a type of data set that may look something like this. So this is a so-called CTA runoff data set. So a large portion of the body is imaged such as this, and this may contain thousands of slices that would then have to be individually screened by a radiologist. This is, for instance, done for diagnosing pathologies such as calcification in blood vessels that may ultimately lead to complete occlusions of these vessels, which is obviously not something that is desirable. And in the simplest sense, this kind of slicing can be done along the three main axes of the volume. In a medical context, there is a certain standard terminology that is usually used. So you have the axial plane, that is this one. You have the sagittal plane that cuts your head in half, and then you have the coronal plane that goes front to back. The coordinate systems can, of course, differ, but there's a rough convention that axial often corresponds to the XY plane in a data set, and coronal corresponds to the XZ, and sagittal to the YZ plane. But that's only a convention, and of course, that can very much be different, and the orientation in the data set may be different. So that is of course very useful, but what is a problem when you're only sectioning your data set along the main coordinate axis is that it can be very difficult to investigate features that are not aligned with one of the coordinate axes. Think about a blood vessel that, for instance, diagonally goes through the volume. then this will be for each of these coordinate axes quite distorted and maybe very difficult to judge whether, for instance, this blood vessel is fully occluded by calcium deposits. So that's why it's very common that to also use oblique slicing, where the slice orientation can be arbitrarily changed, and this is not restricted to the primary coordinate axis. And this is often used in a setup that is called multiplanar reformation or reconstruction. Both terms are used, where these individual slice views are linked together and can be arbitrarily oriented. And maybe it's best to quickly show you a demonstration of this. So here, this would correspond to a typical MPR setup. So you see, I have the three main axes here, and here actually I see the slices in sort of a 3D view. So now you can see that if I'm moving the slice here, the other, or if I'm moving this cursor here, the other two slices move, and then essentially moving the intersection point between those three slices in 3D, such as this. And now I can also, for instance, change the orientation of the slices, and it's usually enforced that then this slice still stays orthogonal to my original slice. And I can drag here, for instance, to then change the position of the slice. So this is actually a very useful and common tool for investigating volume data. Next, we can also look at creating slices that are aligned to particular features in the data. And there are many different approaches tailored towards specific applications, such as investigation of blood vessels, of the spinal cord, many different types of specialized methods exist. As I said, blood vessels are particularly common, and of course this requires that we have first, in a pre-processing step, extracted a center line of our blood vessel. And if we have done that, we can actually go ahead and create a curved slice that is aligned such as this, that will actually always follow the center line. And we can also look at this in 3D. So this may be a blood vessel that we have analyzed, and then we will create a curved slice such as this. This is also called curved planar reformation. But of course, this is not how you would actually look at this in practice. In practice, you would rather say you then keep the slice or the orientation of the slice constant, and instead just rotate around the center line of the blood vessel, such as this. And the advantage of this is that now with one rotation of 360 degrees, you can inspect the entire area within the blood vessel, usually called lumen, to see is there any pathological change there. And as I said, there are many different variants of this. There are approaches that also straighten the blood vessel, such as that it's always in the center. There are particular techniques that, for instance, try to handle or do handle bifurcations, meaning branching between vessels and so on. Now, next, of course, we want to find out, well, how can we actually create three-dimensional depictions from our three-dimensional data? And one very common approach to do this is called volume raycasting. And the idea is here that we have our virtual camera set up, eye point, and we are shooting rays from this eye point through the image plane for each pixel, and then we are sampling the volume at regular intervals, and then combine these values in order to determine what the color of the pixel will be. And there are different ways that this can happen. One of the common approaches is often referred to in particular in the biomedical context, as direct volume rendering. And basically what we're doing here is we are using a simplified model of physical light transport in a participating medium. So you're basically thinking about the volume consisting of many, many small particles, and the density and the color of these particles is influenced by the values of the original field. And this translation of the values of the original field to these colors and opacities, meaning this optical density, is performed using a so-called transfer function. So for instance, if we here on the x-axis have a data value and here have opacity from 0 being completely transparent to 1 being completely intransparent or impenetrable optically, then we could, for instance, say we have a data range here that we assign a certain color, and then maybe we have another data range here in addition, and maybe this additional range here to give us this semi-transparent three-dimensional rendering. So that is in principle very useful, and it's in fact motivated by an approximation of physical light transport. The problem is that the actual design of these types of transfer functions can be very complicated in practice, because if you are specifying a too high opacity for a particular data value range, you will necessarily occlude other areas in the data that you may be interested in. So it can be quite complex to handle. This is why in practice often simpler approaches are used, and one of these approaches is called maximum intensity projection. So here the idea is that we are always showing maximum value along the viewing ray. And the reason why this makes sense is that there are many acquisition modalities where this that are specifically designed such that the regions of interest in the data actually have the highest data values. So what is, for instance, very common is that you are injecting contrast agents into the bloodstream of the patient, again in the investigation of blood vessels in particular, and those contrast agents improve the contrast of the lumen of the blood vessel to the surrounding tissues. And this is why in such cases you often only need a simple contrast adjustment. In many biological acquisitions, for instance, you're using fluorescent proteins that can be, for instance, by genetic manipulation, made to be tied to particular tissue types, and then again you can image these, for instance, optical sections, and the particular types of tissue that you're interested in light up due to this protein, and again will give you the highest data value. Now, this window level specification that I just mentioned simply means that we are saying we are defining a level at the center here, and another range that is this window, and then we simply have a linear mapping from black to white of the data value range to colors, and that will create you an image such as this. Now, when we are comparing these two approaches, we maybe often see something like this. DVR creates something quite colorful, whereas MIP creates these black and white images. But in reality, of course, when we are looking at the same type of transfer function, then we may rather compare those two things. So practically compared, we have a physically based approach with a practically motivated approach, and ideally, you would actually have the advantages of both of those, because by simulating light transport, we get additional cues about the spatial relationships in the data set. But on the other hand, we would like to have the simple control of window level and avoid the spatial ambiguities that you get from always looking at the maximum, because the maximum does not respect any orientation, for instance. And in fact, there is a way to do this. There's a hybrid between those two approaches, where in essence, we can go ahead and create something that essentially follows the paradigm of DVR and but incorporate some aspects of a maximum intensity projection. I do not have the time to go into full detail of how this works, but I will show an example that maybe illustrates this. So here we are transitioning from DVR via this hybrid method to maximum intensity projection. So you see here, we now very clearly see the metal parts that were inserted in this reconstructive surgery. And here we now see that we are losing some of the shape cues. In particular, when we are now stopping the video, can be quite confusing to judge what is in front and behind in the maximum intensity projection. Well, here's another example. Again, we see here that when we are doing DVR, that we of course have occlusion by other parts of the volume, whereas when we are now doing this hybrid method, we for instance see that this unfortunate patient is suffering from a rather large aneurysm. But we can nicely judge the size and the curvature and also the location of this aneurysm, whereas when we are transitioning to pure MIP, we see that this is much more difficult because we're losing some of the shape cues. With that, I'm almost at the end. I think this was a bit of a tour de force, because obviously this is a very large field with decades of research behind that. And I just tried to convey to you some of the principles. Nonetheless, I at least want to mention some of the more advanced topics that have been investigated in this context, such as illustrative visualization, where the idea is that you try to develop ways to visualize volume data inspired by traditional illustration techniques, including effects such as cutaway views and ghosted views or exploded views. You see here, for instance, a volume data set that is visualized with different individual styles. And then, of course, there is also a branch of research that investigates how to use more advanced illumination models. There are certain applications where this can make sense. Maybe medical diagnosis, for instance, is not one of them necessarily, but there are other approaches where it can be helpful to, for instance, get additional cues to do shadows. And you see some examples here where, for instance, you're seeing refraction simulated. Here you see an approach that uses multiple scattering of an actual 3D ultrasound data set to simulate the appearance of fetoscopic procedure, where an actual endoscope is inserted into the uterus. You see very nicely that each of the twins here has its own little chamber, which I find very fascinating. Then, of course, there is a very large area that investigates I would say transfer function spaces, where you can use additional derived quantities for better differentiation of features of interest. So for instance, you can use the first or the second derivative of your scalar valued function in order to characterize regions that you cannot distinguished by the scalar value alone. And then there is also active research on multimodal or multivariate volume data, where you have either data from multiple modalities or different variables, different quantities for each point in space. And then there are things become much more complicated in terms of perception. Mapping multiple quantities to colors at the same point in space can become very difficult to interpret due to color mixing. But also there is additional additional occlusion, and of course it becomes more difficult to specify such a transfer function at all. And of course, there are many more topics in this area. And I hope I at least gave you a short impression of some of the basic aspects. And I will now hand over to Michael, who will explore some more applications of volume data. But I'm happy to answer some of your questions now already, if there are any.
We can't hear you, Ingrid.
Thanks. So there's one quite obvious question for you, Stefan. Is there a place where you share your source code?
The place with respect to what, if I can, if I may ask. So obviously, what I've been talking about here is not just research by me, and for several of our methods there is source code available. We don't have a very centralized repository at this moment, but for individual methods you can find links in the paper. So if you have a specific question with respect to that, I'm happy to figure out whether there's a source code available. For some of the methods that I've shown here that were done in collaborative research with companies, for instance, source code is not available, but also several of the methods that I showed, such as this hybrid meter representation, have in fact been implemented by different vendors from the available description in the paper.
Hey, there's another question concerning the MIP. How do you decide on the best ray direction to collect the maximum intensity?
Best ray direction to collect the maximum intensity. So basically, typically what you are doing is, or what is typically done is you're really looking at the ray direction from the point of view of your virtual camera. So if we are looking at something like this, I can maybe switch to a volume rendering like this, and we can now maybe just specify a transfer function. So basically, here we now have an orthographic projection. So each of our viewing ray, or each of our pixel corresponds to one viewing ray that traverses the volume. And so then the viewing direction is, or the the direction for the maximum is specified by the rotation of the camera here.
For now, I only have one more question, even though there are more, but I think we should restrict it to a brief answer to what do you think is the future of volume rendering?
Oh, that is a very interesting question. So obviously also afforded by the advances in GPU technology. There has been quite some progress with respect to using more advanced optical models, and of course things like real-time ray tracing features on current GPUs can also be exploited for volume rendering. Whether or not full path tracing is something that is computationally justified given the application very much depends on the particular task that you have. I would say in most applications in the biomedical context, there's no need to perform path tracing. But when we are talking about the rendering of volumetric phenomena in general, such as for production rendering in terms of communication to the general public, then obviously things like volume path tracing are becoming more feasible. And in fact, those are at the moment sort of the best of class methods in terms of realism, but they are not still yet at the level where this could be done fully interactively.
So thank you, Stefan.